The retina is made up of several types of cells, all located in the neural layer. These cells convert the input entering the eye in the form of light into an action potential which is sent to the visual cortex for interpretation. In a counterintuitive manner, the light receptor cells, either rods or cones, are pointing toward the pigmented layer of the eye instead of to the cornea. Both types of cells have an outer segment made of many discs which contain the photosensitive units. Towards the lumen of the eye follow the inner segment, the body, and the axon of these photoreceptor cells. Their axon connect to bipolar interneurons, which are interlinked by horizontal and amacrine cells which modulate the signal. Closer to the lumen still are the ganglion cells whose axon form the optic nerve. In darkness, the photosensitive cells located within the disc have cyclic GMP attached to the sodium channel, and as a result, these channels are kept open. Thus, sodium is constantly entering the photoreceptor. At the same time, sodium is being pumped out of the cell by an ATPase pump. This situation maintains a membrane potential of about minus 30 millivolt, which implies that these cells are depolarized. While depolarized, these cells release the neurotransmitter glutamate, which inhibits the release of neurotransmitter by the next cell in the chain, the bipolar interneurons, to the ganglionic cells, preventing the ganglionic cells from sending an action potential to the cerebral cortex. To generate an action potential, light entering the eye hit a yellow pigment called retinal, which is tightly associated to a molecule called opsin. Opsin is part of a larger structure called rhodopsin, which is a protein with several transmembrane domains. The change exerted by the light on retinal causes a cascade of conformational changes in the rhodopsin, which in turn activates an intracellular G protein, which then activates a cyclic GMP phosphodiesterase enzyme. This enzyme converts the cyclic GMP bound to the sodium channel into GMP. When the sodium channel is bound to GMP, it closes its gates, preventing further entrance of sodium. Nevertheless, the ATPase pump continues working, taking sodium out of the photoreceptor cell, and by doing that, it hyperpolarizes the photoreceptor. While the photoreceptor cell is hyperpolarized, the secretion of inhibitory glutamate is reduced. Not being inhibited, the bipolar cells are free to release their neurotransmitter, which stimulates the ganglionic cells to generate an action potential which travels to the cerebral cortex with the information associated to the input in the form of light and color received by the eye.